Welcome to this biochemistry video on protein interactions. We're going to discuss ways that proteins bind ligands, and the ligands can be, ligand is the term, the binding partner. Ligands can be other proteins, ligands can be small molecules, things like drugs or steroids, hormones, even small ions, and so we'll see those very commonly. We also bind DNA and RNA, so we'll talk a little bit about each one of these. When we talk about binding, the number one property that we're going to address, that we're going to commonly discuss, is the binding strength, and that's usually done with Kd, which is the equilibrium binding constant. And so at the top we have some examples of proteins and ligand, the strength of that bond. So for instance, it ranges from avidin, where the protein with a biotin of ligand with a ligand of biotin. So avidin and biotin have a very, very strong bond. It's a 10 to the minus 15th, which is in the femtomolar range. So that's a very, very strong bond. And all the way up through calmodulin, which is in the micromolar range, 2 to the minus 5th. Tens of micromolar range, 10 to the minus 5th. And you can, at the bottom, you can see general ranges. So antibody antigens are typically picomolar up through up to almost micromolar. Enzymes are micromolar up to millimolar, so on and so forth. And so you get an idea of the bonding. This will give you an idea of the range of binding strengths that occur with different ligands. We're going to discuss DNA binding, and we'll discuss it more as this is just an introduction right now. A DNA binding domain is an independently folded protein domain within a protein that has a structural motif that recognizes double-stranded or single-stranded DNA or RNA. And usually it's very specific to the DNA sequence, and that gives it the opportunity to be very, very specific. DNA binding domains usually involve things like replication, repair, storage, modification, blah, blah, all those kind of typical things we've already discussed are centering around central dogma. And these commonly are involved in regulation of gene expression. And so there's about 9 or 10 common ones, things like helix turn helix, which we talked about, zinc finger, leucine zipper, winged helix, helix loop helixes. Some of those are the more common ones. And I was shown here is a leucine zipper, where the protein sits right inside the major groove of DNA, and that provides the binding, and thus we get the structure there. And then the function is usually something like gene expression. So that's a very common mechanism, common way that protein DNA interact, and it's through commonly these motifs that are DNA binding domains. So protein-protein interactions are a very, very important interaction biologically and biotechnologically and pharmaceutically. These are common drug targets to block protein-protein interactions. They're also used in biology for signal transduction, cell metabolism, muscle contraction. They're often enzyme spots, and they're also very structurally related. And they reflect these interactions occur on the surface, and they're very commonly very, very hydrophobic residues in the interaction sites. So it's a great way to identify them, and they're pretty flat and large, although there is some shape to them. They're generally flat, but there's localized regions of topographical features. They're roughly 750 to 2,000 square angstroms. And then again, hydrophobicity plays an important role in this, and that's what you see on the figure over here. So hydrophobicity plays an important role through this concept of solvent accessible surface area. So what we're trying to do is we're trying to reduce that surface area, and that site, that hydrophobic site, will push out the water, and that binding pocket, it provides an important core. That binding site becomes a core where solvents can't interact, and that improves the ability to bond. There's also this concept of complementarity, where when something, we'll discuss it later in the lecture, but this is very, very common with protein-protein interactions, especially things like antibodies, where when something binds, it changes a conformation of the protein, especially of the protein, not the ligand, but both are possible, and that allows it to that change in conformation. There's an allosteric change in the protein, which is basically it changes shape, and that change in shape allows it to perform a different function that was not part of the original conformation of that protein. So again, some other things that occur around this protein-protein interaction site besides hydrophobicity, we also have, again, localized shape that provides like a lock and key type mechanism, and we also have electrostatic interactions that occur at these interactions. These protein-protein interactions can also be polymeric and things, but they also can be just simple dimers, trimers, and things like that, pentamers, tetramers, etc. For pharmaceutics, protein small molecule interactions are important, and they also have spots that are very, very important. Usually, enzymes are the target of a small molecule, but protein-protein interactions on things like hotspots. Hotspots are where important binding sites within a protein-protein interaction, and a small molecule can either block that hotspot, or sometimes it can be an agonist, and that agonist can cause the increase in the function of that molecule. So hotspots are also very tight, small pockets. The surface area binding is significantly smaller. It's in the tens to hundreds of angstroms versus, hundreds to thousands of angstrom size, where these small molecules come in and fit. Things like steroids also fit in these things. And the interaction site is not just a binding site, it's commonly referred to as the hotspot. And hotspots are important ideas is an important idea in improving the ability to deliver drugs, because small molecules are an important drug class. Now, again, we'll see a resurgence of small molecule drug classes. This is kind of the important concept surrounding protein small molecule interaction. We'll also see this with things like steroid receptors and other spots where these are commonly found on the surface, although sometimes in things like GPCRs, which we'll talk about later in the semester when we talk about membranes, those things have a core inside that on the surface of a cell that allow for the binding site to occur. So again, this kind of gives you a visual representation of what we're talking about. Proteins with druggable hotspots. Usually these things are a little bit flatter. An enzyme has a deep binding pocket that the drug can fit in and block that enzyme. So that's kind of the properties that you're looking for. So when we're talking about a protein-protein interaction, it's relatively flat compared to an enzyme binding pocket, which commonly is deep, and it has a very, very tight fit with what the reactants are. All of these interactions again go back to our non-covalent interactions chart, and I don't want to spend a great deal of time talking about them. There's a reason we had to spend an entire lecture. All of these interactions are non-covalent interactions. There are a few exceptions, but generally speaking, these are all going to be you're going to see these things over and over and over, whether we have an ionic bond, a hydrophobic bond, a hydrogen bond, or van der Waals bond, or some pi bonds. Those are the general properties in which you're going to see at binding sites. And commonly the charged amino acids are going to be, or charged or polar, are going to be found at those sites. So there's a couple different models that exist. That we're going to go over two of them. There's the lock and key, where you have, and that's what you see in A, where you have a ligand that's very specific and it only fits inside that. The ligand is very specific and it only fits inside that protein. You also have the induced fit, where you have a site that's not quite the same shape, but once the ligand starts to interact with the protein, you have a conformational shift in the protein, so it changes shape, and then all of a sudden that allows it to bind more tightly. Both of these are very, very common. Complementarity is what you see in kind of B, where you can have complementary in size, shape, charge, and the hydrophobicity of that. So things can kind of change conformations, the size, the shape, the charge, all of that kind of stuff. All those kind of properties can change and allow for specific functionality to occur within these protein-protein protein-protein interactions. Because it's important to remember that these protein-protein interactions are not just in a vacuum. They commonly are used to regulate events or to do work within a cell, and so they are for signaling all kinds of other mechanisms. So these models are pretty important. This is the first introduction that you're going to see to these two models. We'll see them again when we talk about enzymes in a couple of weeks. Lastly on this topic is the biosensor. And when we talk about biosensors are ways we can characterize these types of interactions. And when we do so mathematically and in real time. We can get a yes/no answer with an ELISA, but a biosensor allows us to characterize the properties of that interaction. There's two main metrics by which we do that: kinetics and affinity. Kinetics are a rate of reaction, and you'll see that in this real time with the small Ka, where you see it binding on. You also can see it in the Kd. So small Kd is the off rate. So Ka is how fast it goes on, small Kd is how fast it goes off. We also have an equilibrium rate, which is the big Kd or big Ka. That's after equilibrium. What's the value? So that's actually a more commonly referenced metric. So when I was talking earlier about the binding strength, I was talking about the big Kd. That's an equilibrium binding constant. Again, equilibrium is at a stable concentration, the end where the on rates and the off rates of that molecule are equal. What is the value of the big Kd? So that's what you're going to see when we talk about those two interactions. It's very, very important that you understand the difference between kinetics and affinity. Kinetics, again, are a rate of reaction, and affinity is an equilibrium constant, and we'll go over greater detail with that later and in other slides. Thanks for listening. We look forward to seeing you in the next lecture.